Hi, good morning, everyone. Good morning, everyone. Hope you had a good coffee break. Thank you all for coming back. My name is Mark. I'll be your chairperson for this session. So online right now, we have Anna Namyatova. She will be speaking on integrative approach to the identification and delimitation of the trans-Palearctic Ligus species. Anna, whenever you're ready.
Okay. Sure. Can I start now?
Yeah, please.
Okay. Thank you. Hello. The title of my today talk is the delineation and identification of trans-Palearctic Ligus species using integrative taxonomy. First, I start with the systematic placement of Ligus. It belongs to the Hyperderose family Myrida. It includes more than 11,000 species within.
Anna, we cannot see the screen with the PowerPoint presentation.
Okay, I'm sorry. Yeah, I forgot this. You see it now?
Yeah.
Okay, sorry. Yeah. So, okay, I will repeat my first slide, and the second slide, Ligus belongs to the largest myrid subfamily, which is Myrina, and it includes more than 4,000 species by now, and within Myrina, it belongs again to the largest tribe, which is Myrini, and within Myrini, Ligus and all similar genera form so-called Ligus complex. It is a very hard group for study. So I think everyone who ever tried to identify Ligus species would agree that it is a taxonomical nightmare. By now, it includes more than 270 species, which is known worldwide. Most probably, Ligus is not monophyletic, and species outside Palearctic do not belong to Ligus. Ligus sensu stricta includes 50 species, which are very common in Palearctic. And some species are pests, and they damage cotton, apples, strawberries, and other crops. So what makes it's so hard to study? First, Ligus species have minimal interspecific differences, which means that the species differ in tiny morphological traits in external view and genitalia. They possess considerable interspecific polymorphism, so many synonyms were described. Many species are polyphagous and sympatric, and they can live on the same plant. Some species are widely distributed, and they might have trans-Palearctic, trans-Nearctic, and trans-Palearctic distribution. The keys to species have been published, but identification is still challenging. And now I think we have to accept that the morphological characters for species delimitation of Ligus are insufficient. So where shall we start where we deal with such a hard group? First thing which comes to the mind, it is the revision of all 270 species of Lygus. But of course, this research is impossible for molecular study, and considering the problems with the morphological revision can be even useless. We can reduce the scope and study the integrative taxonomy of Lygus sensu stricta, which includes 50 species. But again, considering the distribution and interspecific variation of those species, this will be extremely time-consuming, and we need to have a proper methodology to make this study effective. We can decrease the scope even more and study the integrative taxonomy of Palearctic species only, and there are 19 of them. This study looks more realistic, but when we look at the map, we will see that the range of distribution is still huge. And among those 19 species, most of them are known from Central Asia only. And it is very hard to collect in this region. In the Zoological Institute alone in St. Petersburg, the collection includes 45 drawers, and it is approximately 100,000 species, specimens. And of course, when we look at those numbers, again, this study looks not so realistic, and again, we need the effective methodology to make this study possible. So we decided to start with the disentangling the relationships of 5 trans-Palearctic species and concentrate on the delineation and identification. Those species are easy to collect and to get fresh material. There are many historical specimens available for the morphological assessment, and we have numerous records for those species which might make the study a bit easier. So for the methods we chosen to study discrete morphological characters, we also studied continuous morphological characters and applied principal component analysis and neural networks analysis. We also included molecular-based studies and used 2 mitochondrial markers, which are barcoding region of CO1 and 16S RNA. It was important to test whether we can use the barcoding region for species identification in Lygus. So the stage 1, the species hypothesis based on discrete characters. We aim to find the characters important for species delineation and to split the specimens into morphospecies. When I sorted the collection and the Zoological Institute, I found that the most of the specimens belong to 5 trans-Palearctic Lygus species. 3 of them are on this slide, and those are 3 species which occur in Palearctic and were not recorded in Nearctic. They are Gemelatus, Pratensis, and Wagneri. And there are 2 more species, the Bulipenis and Punctatus, which have Polarctic distribution. As you can see, it's still possible to identify some species by color. For example, Pratensis have those red markings and red wings, but each of those species have pale morphs, and it is impossible to identify those morphs by color. So what else we can use for the species identification in Lycus? This are the combination of the punctation and seti density. In Gemelatus and punctatus, punctures placed far from each other and seti are very rare. In Tratensis and Wagneri, punctures placed close to each other and seti are denser. And in Rubu Alipenis, the punctures are almost adjacent to each other and seti are so dense, so wings look matte. As you can see, those states are not unique, and sometimes it's very hard to choose between the states which belong to the Bulipenis and Wagneri. So another character is the spicule in vesica shape. In Gemelatus and the Bulipenis, the spicule is tapering, and in Tratensis, punctatus, and Wagneri, the spicule is widened. Again, those states are not unique, but in Pratensis there is another character, which is the absence of teeth on right side of small lobe of vesica, and this is the unique state for this species. So, and the conclusions from the stage 1, 5 transpalactic species do form the morphospecies. In most of the cases, species differ in combination of color, punctation, and vesica spicule shapes. Sometimes females cannot be separated. Those characters are variable, which makes the identification process challenging. And the logical question we have are those morphospecies natural groupings or variation of those characters is intraspecific. And we test these morphospecies using the continuous characters. We apply the principal component analysis and neural networks. Principal component analysis, it showed that all species significantly differ, at least in one sex, except for one pair, which is the Bulipenis and Gemelatus. And we used 22 algorithms for neural networks, and we found that there are algorithms which differentiate each of those species with a 100% accuracy. So the conclusion from the stage 2, the 5 transpalactic Lygus species are delimited using principal component analysis, except for the Bulipenis and Gemelatus and neural networks. And 5 transpalactic Lygus species can be identified using neural network algorithms. And the stage 3, it is the searching for characters in DNA sequencing. In this table, you can see that there are unique substitutions in 3 species, Gemelatus, punctatus, and the Bulipenis. And in Pratensis and Wagneri as a group, there is a single unique substitution, and each of those species does not. And we also tested the monophyly of the species using phylogeny, and we use the 5 automatic methods for the species delineation. And we found that the phylogeny forms 2 well-supported clades. One of them includes palactic-only species, which are Gemelatus, Pratensis, and Wagneri. And the second clade includes Olactic species, which are the Bulipenis and punctatus, and 2 nearctic species, Gisperus and Linealaris, which we included as outgroups. And the automatic species delineation supported this split. If we look closer into those clades, we will see that within the Palearctic only clade, the species do not form the clades. And within the clade, which includes Palearctic and Nearctic species, only Punctatus forms a clade, but the Bulipenis does not. So the conclusions from the stage 3, Gemelatus, Punctatus, and the Bulipenis have unique substitutions. Pratensis wagneri as a group has unique substitution, but each of those species does not. Phylogeny has two well-supported clades. First of them, Gemelatus, Pratensis, and wagneri, and second includes Punctatus, Bulipenis, and Nearctic species. And among all 5 species, only Punctatus forms well-supported clade. And the main conclusions, each species can be separated in combination of morphological and molecular data, and barcoding region alone cannot be used for those purposes. All species can be identified using neural networks, and there are at least 2 monophyletic Lygos species in Palearctic because there are 2 distinct clades in phylogeny, but this requires further tests using the nuclear DNA. So those are my acknowledgements, and thank you for your attention.
Thank you, Anna. So we have some time for questions. Does anybody have questions? Anybody? Okay, we have a question.
Hello, Anna. This is Christiane. I'm not sure if you can hear me, but we may have to, redo the question directly in front of the laptop, maybe. So, here my question. So, do you think Lygos is, generally speaking, completely oversplit, and there are actually many, many fewer taxa, species-level taxa? Oh no.
I can hear you.
Cool. Now we can hear you now.
Da, yes.
Thanks.
Do you want it?
Yeah.
You need mine?
No, it is.
Hi, Christian.
I have a quick question. My question is, given your findings, do you think this may be true for the rest of Lygos as well, that it may be just very oversplit?
Yes, I would say that I also didn't include into my presentation, but I also built the phylogeny with all the species from US and Canada based on the CO1 only. And I found that it doesn't work for those species at all. So I think we need to use more molecular markers to find what is going on there.
Thank you.
So thank you, Anna. So we have run out of time. We will move on to the next presentation.
Okay. Thank you very much.
Hello? Hello?
Hi there.
Can anyone hear me?
Yeah?
At least see me?
Yeah.
Okay. That's good.
So for our next presentation, we have Veronica Tyts, who will be speaking to us on phylogeny of Rhinoceleptus complex. Veronica, you can start.
Okay, can you see my presentation?
Yes.
Yes.
Great. So I'll start. So my name is Veronica Tyts, and I want to present you our research in phylogeny of the Rhinocelepus complex. So, Rhinocelepus complex is from Silopina, which is a mostly tropical and subtropical subfamily within the highly diverse family Murida. Even though biological data for most of the Silopine text is unavailable, close association with fungi or even mycophagy were shown for many species. This subfamily is relatively small, comprising about 500 described species, and quite rare in collections, partly due to cryptic habitats such as rotten logs or decaying wood. Over the last decade, numerous new Silopine texts and several generic and tribal revisions have appeared. However, the monopoly of the subfamily and most of its subgroupings and their interrelationships remain questionable. Currently, Silopine includes 2 large tribes, Fulvini and Silopini, and 3 small tribes, Botremirini, Vaniini, and Trinimirini. Additionally, Silopina is either treated as a separate subfamily or included in Silopina as a 6th tribe. So the Rhinoceros complex is a small group within the largest Silopine tribe, Fulvini. At the time we started our research, it comprised 23 species from 7 genera. It's characterized by external efferent system of metasthoracic glands, which has a reduced evaporative area with a flat peristome, a sparkle without microsculpture, characteristic parameter shape, and 2 large symmetrical sclerites on posterior wall of bursicoplatrix. Also, most of the species of the complex are sexually dimorphic. Males have curved second antennal segment and spines on labrum, and they are also usually 2 times smaller than females. Yasunaga observed interesting mating behavior in one of the species of the group. Male rode on the final female instars, holding tightly by second antennal segment and labral spines. Those horsemen sat on the chosen female larva until it emerged and then copulated with general imago. So let's meet all the genera of the Rhinoceros complex. I'll start with Punctifolius, the most different from other genera of the group. It has a unique, deeply punctate body and a rounded pit between calli. This genus has short head and isn't distinctly sexually dimorphic. The first label segment subdivided with suture is also an important character of Punctifolius. This isn't unique in Fulvini, however, in all other representatives of Rhinoceros complex, this segment isn't subdivided. Preamble is too characterized by short declivous head and the absence of distinct sexual dimorphism. Its unique feature is curved second label segment. The next one is Mesotocylapus. This genus, as well as all other genera of Rhinoceros complex, except for Punctifolius and Preamble, show distinct sexual dimorphism. It's characterized by having a small evaporative area and calli occupying half or more pronotum. Head in Mesotocylapus is small with rounded fronts. Eumetophobus, too, has short head. However, in comparison to the genera I listed before, the head has straight front. So we've got 3 species left, all characterized by having an elongate head with straight fronts. The first one is Rhinoceros, different from other in the second antennal segment, bent or only slightly curved in males. The second one is monotypic genus Rhinoceros, which was actually synonymized with Rhinoceros, but Wolski restored its status. It's clearly distinguished from other genera by its elongate oval body, slit-shaped pit between calli, and broad, punctly punctate embolium. And the last one is Rhinocelopoides. It has shortened wings in males, and similarly to Mesotocylapus, its calli occupy more than half of the pronotum. As we now know the players, it's time for a bit of history of the complex. So Rhinoceros complex was first recognized by Gachista in 2000 within the tribe Trinimirini to accommodate only 3 genera, Preamble, Rhinoceros, and Rhinocelopides. In 2010, Wolski first revised the Rhinoceros complex, gave an exhaustive diagnosis of the group, and added Mesotocylapus in it. Then, in 2011, Volsky and Garchitsa described the new genus Rhinocelopoides, which they assumed to be close to Rhinocelopus and Rhinocelopides due to the elongate shape of their heads. The next work concerning the complex was by Namyatova and Cassis in 2019. It presents the first phylogeny for the Rhinocelopus complex and actually the only molecular and total evidence phylogeny for the subgeneric celopine grouping. The main aim of that study was to test the monopoly of the tribe Rhinomerini, which at that time included Rhinocelopus and Rhinomeris complexes plus Rhinomeriella. Their results showed that Rhinomerini is not monophyletic and its subgroupings are not related. In particular, the Rhinocelopus complex and Rhinomeriella were placed within tribe Fulvini separately, and Rhinomerini was restricted to the Rhinomeris complex. Phylogeny presented in the paper recovered Rhinocelopus complex as monophyletic with strong support and with Punctifolius as sister to the remaining genera. They also added two genera, Punctifolius and Nematofolius in the Rhinocelopus complex. However, the phylogenetic relationships of other Rhinocelopus complex genera were either unresolved or had weak support. And the molecular data was present only for 4 species of the group, and the authors did not find a strict synapomorpha for the complex. Additionally, the position of the Rhinomeris complex required further testing, as molecular data was present only for 1 specimen of the group. So, with this work, we aimed to provide a detailed phylogeny of the Rhinocelopus complex, and during it we also found a new monotypic genus from Borneo, which we aimed to describe. This research could not be possible and actually would not have been started without Klaas Duncan and his unique material which he collected in Brunei Darussalam and Borneo and generously sent to us. Oh God, I've lost. Oh, yeah. With that, we managed to obtain molecular and morphological data for the new genus and several other representatives of the Rhinocelopus complex. And we significantly supplemented the data of the first phylogenetic analysis of the group performed by Namyatova and Cassis. In total, we studied more than 50 specimens and obtained DNA data of the 4 markers for 6 more species. We constructed 7 phylogenetic topologies using the methods of parsimony, maximum likelihood, and Bayesian inference, which were based on an expanded morphological matrix with 65 encoded characters for 38 species, representing 16 species of the Rhinocelopus complex, and molecular data for 23 species, representing 7 species of the complex. So I'll start by presenting the new genus. It's called Rhinocelopidae. Okay, we're probably not that cruel, and it's named Tatupa and contains only 1 species, Tatupa graphi. Although being externally extremely close to Preamblia, with only minor differences such as more sparsely punctured pronotum in comparison to Hemelytron and distant cavern lateral in pronotum, Tatupa has a unique, strongest, entirely stereotyped C-shaped vesica, totally different from what we see in Preamblia, and other representatives of Rhinocelopus complex all have a membranous vesica. The molecular data also confirms the generic status of Tatupa, which we'll see a bit further. So, we managed to obtain a more resolved and consistent topology. However, our results were mostly congruent with those presented by Namyatova and Cassis. In particular, we confirmed that Rhinomeriella, Rhinomeris, and Rhinocelopus complexes do not form a clade. Rhinomerini sent to Namyatova and Cassis is monophyletic, and the Rhinocelopus complex and Rhinomeriella are placed separately within Fulvini. All topologies render Fulvius as a sister taxon to the Rhinocelopus complex, and Punctifolius is shown to be sister to the remaining taxa of the complex. Within the Rhinocelopus complex clade, the morphological analysis showed the same result as morphology only in total evidence analysis in Namyatova and Cassis. whereas the molecular and combined total evidence analysis show additional differences in comparison with the previous study. First, we added data on the monotypic genus Tatupa, which we can see is placed either as a sister clade to Preambly in analysis comprising morphological data, or groups with Rhinocelapus forming a sister taxon to Rhinocelapus vittatus in all molecular data only analysis. Second, in the Metavencasis, the position of genera within the Rhinocelapus complex was not clear and varied between all analyses based on different data sets. Our analysis shows Mesocelapus placed as sister to Tatupa, Rhinocelapus, Preamblia, Rhinocelapoides, and Rhinocelapidius on all trees except those based on morphology only. The morphology-only tree we have seen, similar to Nematovencasis, shows Rhinomatopulius and Mesocelapus forming a clade with Rhinocelapus, Rhinocelapidius, and Rhinocelapoides, as all of them share distinct sexual dimorphism, while Preambly and Tatupa are sister to the above listed genera on that topology. Third, the position of Rhinocelapus vittatus was unresolved in the Metavencasis, and the results are the same for the morphology-based analysis in the present study. The results of our analysis using molecular data cast doubt on monopoly of the genus Rhinocelapus, as Rhinocelapus vittatus does not form a clade with other representatives of Rhinocelapus genera, and is either placed as a sister taxa to Tatupa in molecular-only analysis, or forms a clade with Rhinocelapidius in total evidence analysis. Rhinocelapus vittatus and Tatupa both share similar sclerites of the posterior wall of Bolshakova, whereas as for the clades of Rhinocelapidius plus Rhinocelapus vittatus, we also found a synapomorphy. Its label segment 4 longer than segment 3. However, that character isn't unique. Therefore, Rhinocelapus vittatus may form a separate genus, as it also differs considerably from other congeners in many character states, including pale brown color pattern, claw with subapical tooth, and well-developed supragenital breach of the genital capsule. In addition, this species does not have spines and labrum, which are present in other Rhinocelapus, Rhinocelapidius, and Rhinocelapoides species. If we've got more material, we could also discuss the problem of the unique synapomorphy for the Rhinocelapus complex. It was mentioned by both Wolski and Namyatova that the characteristic shape of parameres could serve as synapomorphy of the group. In species of the complex, the right paramere is almost straight with conical outgrowth near apex and widened in the middle, whereas the left paramere is strongly curved and dorsally elongated on the apical part. The Metavencasis also indicated that the presence of two large symmetrical sclerites in the posterior wall of Borsacopolatrix in female could also be its unique character. The results of our study support the assumption that the shape of the parameres of the Rhinocelapus complex serves as its unique synapomorphy. However, the two large symmetric sclerites are not a unique synapomorphy of the Rhinocelapus complex, since such structures are also found in other Fulvini, for example, Fulvius species. And instead of the conclusion for this study, I wanted to ask, has anyone seen this species? We found only one female specimen of it, and I guess it feels lonely and really hopes for description. Thank you. I'll be happy to answer your questions. Has anyone heard anything?
Thank you, Veronica. Anyone has any questions? Any questions? No, no questions. So thank you, Veronica.
Thanks a lot once again. Bye.
next presentation, we have Minsuk Oh on the molecular phylogeny of the family Miridae with discussion on divergent state and life history evolution.
Good afternoon, my name is Minsuk Oh from Seoul National University, and today I will talk about molecular phylogeny of the family Miridae with discussion on divergent state and life history evolution. My presentation will go through these 4 parts, and let's get started with introduction. Family Miridae normally distinguished by diverse body size and for segmented antenna and labium, existence of cuneus and lack of ocelli except subfamily Isometopinae, and in genus and species or tribal level, pretarsal structure and genital structure are also regarded as important key characters. Their taxonomic position is the one of the family from improved Simicomorpha and includes 7 or 8 subfamilies depends on its addressing of Psilopinae, and also known as the most species rich in Heteroptera, which includes approximately 11,700 species. When we see this pie chart, diversity of Miris shows exceptionally large when comparing with other higher groups like aquatic insects or Pentatomomorpha or Simicomorpha without Miridae. Miridae occupies 1/4 of entire Heteroptera diversity. As in their diverse morphological characters, their biological traits also shows large variation. Their habitat is mainly on plant species, plant tissues, but sometimes on tree barks or fungi like Silapinae. And among plant species, some species resides at Eudicus or like other plant groups, fern or coniferous trees. And sometimes their feeding habits are fungivorous, phytophagous, and predaceous, but mixed with each other, like mainly fungivorous, but sometimes predaceous, like in facultatively, or mainly predaceous, but sometimes use plant source as their energy. For that, their diverse biological characters and morphological diversity, some people try to definitize their internal relationships. In this 4 cladogram shows beforehand studies based on morphological analysis, and we can see normally Isometopinae places as most basal group, and sister group relationships are shown at Deriocorinae, Myrinae, and Orthotrinae and Pyrinae respectively. And Bryocorinae and Silapinae normally shows monophyletic nature in most recent cladograms, in morphological cladograms. And when started of 21st century, phylogenetics works based on molecular and total evidence works are also conducted by Xu et al. And Zhang and Li. Their topology shows large difference between morphology-based works, like basal group position changed from Isometopinae to Pyrinae, and sister group relationships of some groups were denied, and Bryocorinae and Silapinae were regarded as largely paraphyletic or polyphyletic groups. These changes in cladogram also leads to changes in ancestor state estimations of Miridae hypothesis. Before this molecular work, largely Miridae's feeding habit origin regarded as predaceous based on physiological and morphological characteristics, and this also supported by background cladogram as predaceous Isometopinae as a basal group. However, if after Zhang and Li, phytophagous origins are newly hypothesized, and background cryogram also shows Phytophagous pyrine as a Bowsher group. And subsequent studies that focused on hemiptera and heteropteran species divergence also shows Myroidea and Miridae largely looks like plant feeder dominance or have phytophagous origin from plant tissues. At the same time, J.R.I also estimated divergence date and suggested first divergence dates on Myriads may started at Permian. However, this results largely discordance between subsequent studies, and this makes hard to make decision how Myriad species will diverge and evolution from goes with prawns or evolution and diversified with angiosperm radiation. This one can be a problem. And when reviewing beforehand studies, we make 4 aims for our studies. At first, we try to reconstruct Myriad phylogeny and with more robust data, and based on this result, we try to make decision on ancestor stage hypothesis, whether they are predaceous origin or phytophagous origin, and also we wanted to suggest additional hypothesis for divergence date estimation, and then last, we also wanted to provide some plausible factors that may draw high diversity of Myridae. To do this, we did following Bessarian method. We improved some species diversity in our cladograms with comparing with beforehand studies, and we tried to make more species, enlarge species number for shortage of certain groups at J.R.I, like bryocorine and silapine and isometopine. In addition, we tried to add some more protein coding gene markers to do our phylogenetic work, and at last we made data set with 7 subfamilies, 38 tribes, and 138 genera of in-groups, and including 11 out-groups, a total of 203 species. This data set analyzed by Architree and Mr. Bass and Pope to get data of maximum likelihood, Bayesian inference, and maximum parsimony, and all results were visualized by Victory. And in ancestor trait and divergence date estimation, largely beforehand studies shows absence of fungible state in 2012 study by J.R.I, and there was lack of habitat estimation just for Myriads. So we tried to rehabilitate that things, and in previous studies, their estimation only for in-group, so we also included fossil data and sequence data for out-group to estimate their divergence state. We use 3 feeding habit estimation traits and 7 traits for habitat estimation like this. And in fossil data, we use 2 from outgroup and 3 for Myriad divergence state estimations on Myriads. And based on this method, we could make final tree like this based on maximum likelihood. And when we can see this, we also simplify this result to merging some clays to tribe level, and shows Myroidea that defined by previous studies were well conserved with 3 families, Tingidae and Thaumastochoridae and Myridae. However, sister group placement was changed from Thaumastochoridae to Thaumastochoridae from Tingidae, and different from previous molecular work, bryocholine trimonoline is stably placed at beta node in our analysis. Also, some groups like isometopine, pyrine, and myrine retain their monophyly, but bryocholine fragmented into 6 distinct clades, and similarly, silapine lost its monophyly in our analysis. In addition, disipine and acrytotartine also lost their monophyly in tribe level. We cannot certain that this result can definitize each group's proper placement, but at least we can suggest maybe silapine and bryocholine could be non-monophyletic groups. And in deriocholine and orthotyline, thalmatopyline and corridoromyline displaced from each subfamilies and construct a distinct clade. This may imply corridoromyline and thalmatopyline these subsequent taxonomic allocations and these new taxonomic status. In tribal level, for some large groups like deriocholine, myrine, and orthotyline and pyrine, also revealed as largely polyphyletic, and this polyphyletic result may derive from their classification because some external character similar species or almost all included into that large tribes in old times or until recently. And in ancestor state estimation based on this result, we got predaceous origin of mirrors, and after that they gradually shift to fungivorous and phytophagous feeding habit, and at that time intermediate feeding habit, mixed feeders were largely emerged. In the point of habitat, they are predominantly revealed as living plant tissue origin, mostly Unicode, but their habitat shift to tree barks and fungi when their feeding habit changes into fungi. And also after that divergence, sporadic divergence into other plants group like ferns, coniferous trees are also observed. I focused on discussion on mixed feeders because they can grant big advantage to plant dwellers. In Young's study at 2003, he used ambush bug, Pymata pennsylvanica, which have feeding habit with predominantly predaceous, but also can use its plant diet source. In his experiment, he shows despite only plant source like squirrels couldn't make them go to adult, but longevity was largely increased compared with water only or nothing feeding environment. We can imply that mixed feeding can give more opportunity of energy source finding to give them more survival rate, higher survival rate, and more opportunity to give find more diverse food source. And in divergence datum period, our result got first divergence date of Viridae as early Jurassic, and after some major subfamily groups divergence date, like Myrine and Pyrine, Orthotyline and Deriocholine, angiosperm radiation period overlapped with this first divergence of large subfamilies. And date overlap occur also between divergence date of subordinate group. This, after red square, the blue arrow, the rapid increase of Unicode, there are show actively diverging of mirae also observed. And when cross-check with feeding habit evolution, maybe phytophagos could be advantage to diversifications on berries. In our results, maybe partly biased, but we can see predominantly phytophagous berries shows high diversity, and some mixed-feeded dominant ornithine and pyrenees shows intermediate diversity, and predominantly predaceous isometopine and dericoline shows relatively low diversity. In summary, we studied with mildew, and found 4 subfamilies were revealed as polyphyletic, and mildew needs additional taxonomic reassessment in tribes to subfamily level. Also, we found mildew revealed as predaceous origin with subsequent emergence of phytophagy. In this process, mixed feeders were emerged, and these mixed feeders made less advantage to plant dwellers. And in divergence test, we found first mildew emerges at early Jurassic, and subordinate groups rapidly diverged with overlapping period of angiosperm radiation. And we suggest anomalous diversity of mildew may derive from angiosperm radiation and emergence of mixed feeding. This is the end of my presentation. Thank you for listening.
Thank you, Minsuk. May I know if there are any questions?
Thank you, great talk. So how do you explain that drastically different time of divergence for Meridae compared between your study and some of the previous study, in particular the 2012 study?
I think main factor of that discordance between our studies and Zhang and Li may derive by inclusion of in-groups or only using inclusion of estimation with out-groups or only with in-groups. Maybe I did my analysis only with in-groups in my laboratory, but they shows result like Zhang and Li's, and when calibrated with outgroups, the divergence dates goes much similar than other studies like recent divergence dates estimations like J.R.I at 2022 or some current results.
Does anyone have any other questions? Yeah, sure.
So the predatory bugs and the mixed feeders, did they just emerge once or several times, and how many? And was this mixed feeding, is it always an intermediate stage when switching to predator lifestyle, or could it happen independently, just from phytophagous immediately to predatory lifestyle, or do they have to go through this intermediate mixed feeding stage?
In my result, some of them goes to predator to phytophagous, or sometimes re-diverged to some predaceous, like in subfamily Myrinae, the ancestors were largely regarded as phytophagous origin, but predaceous also later re-emerged at some genera like Philostepanus or some predaceous dominant genera, as I know. Yes, for that their feeding habit is very largely changed or re-derived or changing, in my opinion.
Thank you. That's all the time we have for questions. Thank you, Minsuk, once again. So for our next presentation, we have Alexander Knudson. Today he'll be speaking to us on cleaning out the junk drawer, a phylogeny of the Telonemia Costa generic complex.
Thank you. Hopefully there aren't any more weird things, but that's my name in white. I'm happy to share my part of my doctoral dissertation with you all. So the genus Teleonemia is the second most speciose genus of Tingidae in the Western Hemisphere, with 86 described species. It's widely distributed from southern Canada to central and even southern Chile and Argentina and throughout many Caribbean islands. It's also found in extremely diverse habitat conditions, from very xeric, dry deserts to temperate grasslands and even tropical and subtropical rainforests. And there are also diverse TOS associations for what's known, spanning Bigononaceae, Anonaceae to Verbanaceae. And several species that feed on verbenaceous plants have been used to control lantana species around the world. Up in the upper right corner, we can see the charismatic lantana lacebug with its fuzzy discoidal cell. And in the lower right corner, we have a generalized phylogeny of the Tingidae and related fossil families. All of the genera that I'm focusing on today are in the Tinginae subfamily and placed currently in the poorly phylogenetically understood tribe of Tinginae. So Teleonemia has a bit of a storied past, but there's been four main entomologists that worked with Teleonemia: Carl Stahl, George Charles Champion, Carl John Drake, and Oscar Monti. And one of Drake's students, Margaret Poore Heard, in 1946 actually attested that the generic limits were distended even then, yet Drake continued to describe species. Here we can see some examples of the genus Teleonemia. At letter A, we have the type for the genus, Costas funera. And you can see that there's differences in terms of the overall body plan for the antennae. Some are shorter and some are quite slender and elongate. The distiflagellomeres can also be rather clavate or extremely elongate and spindle-shaped. And the pronotal hood can be extremely low and very collar-like, or it can be even in Morio, almost like a small tumid hood. And additionally, the hemelytra can vary from extremely narrow to quite broad. So just a general breakdown of the history. Costa described Teleonemia in 1864. Shortly thereafter, Stahl described the subgenus Amerostyrphus for the genus Tingis for his species Morio. And then in 1873, Stahl described the genus Europharsa, and then divided Tingis into four subgenera, two of which are Melanoropalia and Americia, and transferred Amerostyrphus to the genus Teleonemia. Then, just before the turn of that century, Champion synonymized Americia and Amerostyrphus into Teleonemia. In 1916, Osborne and Drake described the genus Alveotingis, and a year later, partially described the genus Hesperotingis, stating he couldn't assign it to Osborne and Drake's genus or any of Stahl's genera. Then Blatchley, in his treatise of Eastern Heteroptera, separated these genera based on the overall shape of the antennal clubs and also the height of the pronotal carinae. And a decade later, Drake and Poore described the genera Personemia and Eulonemia to for species that were previously assigned to teleanemia that are found in the Eastern Hemisphere. And then again, in 1946, Hurd attested that many of these genera were related, but separation, at least of teleanemia, was quite difficult based on how variable the characteristics were. And there's been literally no work done determining the generic boundaries to present day, or testing them. So here are some examples of the related genera. And you can see here that the antennae can vary from extremely broad, elongate and slender, and some of which even have clavate apices. The distiflagellomeres can also be elongate and spindle-shaped, or even obclavate. And the Hemelytra, some species can vary from being brachypterous and intermediate forms, as well as fully macropterous, and the costal areas of the Hemelytra can vary from quite narrow to extremely broad. And I know not all of us here are taxonomists, so we can kind of apply some of these principles to our daily lives, right? So I'm sure most of us have a junk drawer at home where we just dump everything in, we don't know what to do with. For example, this drawer of kitchen utensils, or they all kind of have handles mostly, and they look like they serve functions, but are they really all related? And as taxonomists, what we try to do is classify things based on their characteristics. So some of you might have a very organized junk drawer with each individual component separated by their individual characteristics. So this lends us to our research questions and overall hypotheses. Is teleanemia monophyletic, given all the diverse forms we can see in one genus? And while we're at it, are the related genera monophyletic as well? So this lends us to our materials and methods. I contacted 92 natural history collections, of which I received 59 loans from 54 of these collections. I was also fortunate to have pre-pandemic visits to the Carnegie Museum of Natural History, the Instituto Nacional de Biodiversidad in Costa Rica, before it was completely transferred over to the Costa Rica National Collection, Texas A&M University, and the United States National Museum of Natural History at the Smithsonian. Whenever possible, I requested and received photographs of the majority of the type specimens, including many that were lost in the fire in Rio de Janeiro. And to date, I've examined over 4,500 specimens, representing 132 in-group taxa, which were included in my analysis. Here's just a simple breakdown of the taxon sampling. And if you remember, I said there's 86 described species of teleanemia. So I've got a few undescribed species I'm currently trying to wrangle. I created a 98-character multi-state character matrix in Mesquite 3.7, and of which 91 of these characters were determined to be phylogenetically informative in PAUP. And here's just the breakdown of the first 13 characters. I did have some binary, but it was really easier to just have multi-state. For the phylogenetic analysis, we conducted a heuristic search with 500 random replications with one starting tree per replication in PAUP 4.0. And the characters were weighted equally and treated unordered. I used tree bisection and reconnection. And herein, we present a 50% majority rule consensus. So it's another big tree that's really hard to see. But this is the consensus of over 6,000 most parsimonious trees. And just based on the morphological data I have alone, some of these nodes aren't really well supported. But I've recovered Alveotingus in orange as monophyletic, Europharsa in purple as monophyletic, Hesperotingus in gold as polyphyletic, Melanoropala in red as paraphyletic, and teleanemia in blue is also polyphyletic. And bisecting teleanemia, that little tiny white space, is one species of Parisonemia. I was unable to include the type for personemia or eulonemia due to limited availability to specimens and loans I requested. So let's take a deeper look at the alveotingis, melanoropala, and hesperotingis clade. So alveotingis is recovered as monophyletic, and that's sister to a clade containing three species of hesperotingis and 2 species of melanoropala, which are Melanoropala sensu stricto, and 2 of which are sister to Melanoropala, Hesperotingis occidentalis and Hesperotingis illinoisensis. These 2 species, based on their morphology and actually really well supported my analysis, I'm considering erecting a new genus. And all of the above is sister to the Hesperotingis sensu stricto. So now let's take a look at teleanemia. It's not as bad as you thought. So we can kind of look at it in about 6 major groupings. Near the base of the tree, there's either a grade or possibly a clade. I'm working on trying to obtain more data to really figure that out. But it's a project I'm currently working on with Tom Henry, and it might represent a new genus. And if you remember, personemia fell in teleanemia in my analysis. However, it differs considerably based on overall morphology of the pronotum, in which in characters that I weren't able to actually use in my initial analysis. So its placement might not belong there, and additionally, it was only supported 62% of the time in all of my trees. Also, Urifarsa fell in one section of teleanemia, and if I were to, if this is actually good, if I could use genes, and Urifarsa falls in the same place, I'd have to erect like 15 or 20 new genera to keep it. So it's probably going to have to be synonymized, or I just need more data. But I'm still trying to figure out the exact generic boundaries based on morphology. And one important characteristic that I found is that the shape of the osteolar paratreme, which is really tiny on this slide, but it's in teleanemia sensu stricto, it is a kind of very small, rounded paratreme that's dorsal margin ends far from the base of the hypocosta. Many other species of teleanemia have an extremely elongate paratreme that's dorsal margin reaches or overlaps with the base of the hypocosta. And I've got other characters that do sort of define these groups, but I'm still waffling whether or not I want to split this up. So, Hesperotingis, Melanoropala, and teleanemia are not monophyletic. Amerosterphus may need to be resurrected from synonymy to accommodate the incongruence within my phylogeny, as well as possibly synonymizing the genus Urifarsa. And the generic complex may also include the genera Personemia and Eulonemia, but further evidence is needed. And right now I'm currently obtaining sequences for select representative taxa to augment the analysis, and I'm also planning on visiting a few natural history museums to further capture more morphological data. About 20 plus species were scored only based on photographs of type specimens, most of which I don't have ventral views. So, and there are very important characters on the ventral surface as well that I wasn't able to examine. Here's just a succinct literature cited. And I could spend eons thanking everyone who's helped, but a huge shout out to all the collections managers and curators that answered all my queries. It spanned 17 countries and 34 US states. And also just a huge thank you for anyone who provided material, sent photos of specimens, and for all the funding that I received to work on this. Anyway, with that, I'd be happy to take any questions you have.
Thank you, Alexander. Do we have any questions? We perhaps have time for one or two questions.
So that one species that was interrupting your strict sense teleanemia.
Yep.
Is that something for which you couldn't see ventral characters? I imagine you would mention that it was the case. But.
Yeah, so that specific species, the overall shape of the pronotum is much more, it's much shorter than anything that's currently placed in teleanemia. And additionally, the paranota are shaped differently. The entire complex, actually, especially teleanemia, the paranota have mostly been attributed to be uniseriate, like one row of cell areoli, but actually if you look at them on the ventral surface, there's a minute row of areoli, and all of these species in this complex have that, and then there's an additional row, but that species of Personemia lacks that as well. So there's two really discrete, easy characters, and one of which, the length of the pronotum, I didn't use, but I did use the paranota one, and it fell within teleanemia, but the morphology is messy. Does that kind of answer?
Well, I guess I'm also curious.
Yeah.
What proportion of your problems do you think are belied in the mystery of not seeing ventral size and type specimens? Because I imagine you have seen specimens of trusted identity of these same species that you could have looked at, or did you not score them?
There were 24, at least 24 species that I only scored from photographs of type specimens, many of which I did not have the ventral view. And there are no photos of any other material because many are only known from the type series. And I have several characteristics on the ventral surface that I was not able to assign, like how the rostrolaminae are formed, coloration, and overall vestiture. And so that led for a little bit poor structure within the tree. So there are a couple taxa that I didn't mention that are also really dubiously placed. So that's one reason why I'm still trying to really figure out the generic boundaries. I've got characters that do work mostly, but I can't confirm for some species.
I'm satisfied. Carry on.
Okay. Do we have any other questions? We have time for perhaps one last question. Sure. I can come down. Oh, okay. All right.
You've already done this job, Alex. Great talk, Alex. It's fabulous to see the tingents getting some kind of play out there. I just got one naive question. Is this J.R.I. species? Any ideas on why it's got this sort of elaboration, of the wings?
That is a really great question. A really great question. I don't know if anybody's even really posited why some genera have extremely broad hemelytra, but there's some anecdotal evidence for certain species that it could actually be some camouflage. A lot of species feed on the abaxial surface of leaves, and they produce an interesting shadow that's much different than something that would be their size and not transparent. So that is something that really needs to be investigated that no one has ever looked at.
Right. So thank you, Alex, for the talk. So lastly, we have Fabiano Stefanello. So our last speaker for this session will be Fabiano Stefanello. He will be speaking to us on integrating time and space to shed light on the evolution of giant water bugs in the New World.
Share my screen.
Yeah.
Start my presentation.
Yeah, you can start, Fabiano.
Okay. So, hello everyone. My name is Fabiano. It's a great pleasure to be here again, but unfortunately this time I wasn't able to attend in person. I finished my PhD last year in November, and recently I got a position of the visiting professor at Maranhão State University in the northeastern region of Brazil. Today I'm going to talk about the first chapter of my dissertation, entitled as Integrating Time and Space to Shed Light on the Evolution of Giant Firebugs in the New World, Mitra Bellostomati Bellostomatini. This work was conducted in collaboration with Professor José Ricardo Inácio Ribeiro. So, Bellostomatini is composed by three genera. Avedus, with 12 species, classified in the four subgenera. Belostoma, consisting 68 species, traditionally classified into 16 species below, and Viberiella, which is a monotypic genus composed by Viberiella rhomboides. As you can see in the figure, the Bellostomatini is exclusively from New World, occurring from southern Argentina to southern Canada. In blue bars are indicated Bellostoma, which is New World wide distributed, in between rivers, lakes, and streams, temporary ponds, but the greatest diversity of species is present in the tropics. Avedus in orange is restricted to the small portion of North America and Central America, especially in Mexico, and in between high elevation springs. Just one single species expanded its distribution to northwestern Colombia. Additionally, this tribe is the most diversified lineage of giant firebugs, representing half of the number of species of the family. And also, they show a great variation of body size, sternal morphology, and male genitalia, which is useful to recognize species and discriminate species group or subgenera. The first time Bellostoma and Avedus were considered a sister group was in 1961 by Lauki Menke, just considering the author's morphology knowledge without any classification method. Almost 60 years later, Ribeiro and colaboradores in 2018 proposed a phylogenetic hypothesis for the family, the Bellostomatini, including all genera, and in this work they recovered Bellostomatini, now including Viberiella, in this case Viberiella, sister group of Avedus. And as the sister group of Bellostomatini, they recovered the tribe Diplonikini. As I said, Bellostomatini is restricted to the New World, while Diplonikini is present in the Afrotropical, Australian, Oriental, Palearctic, and Sino-Japanese region. So, based on the diversity of species of the tribe and the phylogenetic gap for understanding morphological and evolutionary issues, we make some questions, such how the lineage of Bellostomatini evolved in time and space. Which historical biogeographic process were involved in generating the current distribution of the species in the New World? Are the genera monophyletic? If I use more samples, how they are related? Are the current subgeneric classifications supported in a phylogenetic context? And which morphological character supports such subgeneric classification. So to answer these questions and carry out this study, I visited many Brazilian and foreign institutions to examine material and perform law of species. Also, I spent 6 months in an internship at the University of Kansas, because the KU Museum holds the most important collection of giant wearables in the world. Thus, we are able to sample 70 of 81 species. About 90% of the species diverse are described. Then 11 of 12 species of abundance, 58 of 68 species of Belostoma, Ribeiro rhomboids, plus 10 outgroups, including the species of other 5 genera of Belostoma, including the Phosphorus, Pistogia, Bolshakova, Namyatova. Also, we include species of all subgenera or species though previously proposed, as well as species from extreme of geographical distribution. So after examining the material, we propose 94 morphological characters based on external morphology and morphogenetera, and 3,173 molecular characters from CO1, 16S, 18S, 20S, and Easton. 14 species was studied based on only morphology because material were very old. For data analysis, we use 5 fossils to calibrate the 4 nodes of the tree. 3 fossils from Brazil, crater formation from early Cretaceous, fossil from Canada, from Paleocene, and another recent, more recent fossil from Pleistocene of California, of the Belostoma bucket. We carry out we perform the node data analysis in Mr. Base using the whole data set, morphology plus mean. So as a result, we have monophyletic genera. Also, we have good support for internal nodes, as you can see. We recovered Viberiella as sister group of Belostoma, differing from Ribeiro's hypothesis. And for Belostoma, we recovered 3 clades. We are formalizing as subgenera to improve the subgeneric classification of genes. The subgenus Belostoma pertustoma, indicated in the letter A, consisting of species exclusive from South and North America. The subgenus Belostoma microbelostoma, indicated in letter B, consisting of the smallest species of the genus, and subgenus Belostoma sensistrito, indicated in letter C, consisting of predominantly species of South America, with medium and large bird size. Also, most of previously proposed subgenera of Abedus and species growth in Belostoma were recovered as monophyletic, indicating in this case that the taxonomy and phylogeny have met. So related in divergence time, we have estimated that the ancestral of Belostoma plus G20 here in this node have split up after the break separation between America and Africa. And the main period to diversification of the lineage of those tomatini is estimated to occur during the Paleocene-Eocene thermal maximum, which was a period very warm and wet in the past. So as I said, Paleocene and Eocene thermal maximum seem to have played an important role in the diversification of the lineage of those tomatini, when the number of the lineage had significantly increased during this time, as indicated by lineage two-time plots showing a deviation from simulated constant for that model. In relation to the morphology, we have mapped the character state on the main tree using combining morphological and molecular data set under Bayesian estimation. And here we map some synapomorphic, like in Abacus. The 40 tibia has the inner part enlarged or very enlarged, while in the Belostoma and the Ribeiro, the 40 tibia is evenly narrow. Metachifus is have the distal part parallel in Abacus, while in the Belostoma this part and the Belostoma is Ribeiro, this part is concave. But the most important character in a generic level is paramere. In Abacus, the hook of paramere, here indicated, is projected distant along the same axis of the paramere, while in the Ribeiro and the subgenus Belostoma pertustoma, the hook is projected in a diagonal axis in relation to the axis of the paramere. And other subgenera of Belostoma, the hook is projected perpendicular to the axis of the paramere. Also, the Paleosoma is quite important. In other species, there is a membranous projection of the gonopora above the dorsal arms of Paleosoma, while in this clade, composed by subgenus Belostoma, Microbelostoma, and Belostoma sensistrictus, the gonopora is there is no any elevation in gonopora. Also, subgenus Belostoma, Microbelostoma, as I said, is composed by the smallest species of the genus, with about 9 to 18 millimeters of body size. Also, these species have few denticles in the lacine compared with other species. The twin groove of Metanotum is rectangular, while in other species is rounded. And in this subgenus, the male genitalia is quite variable, from a very spatulated and striped diverticulum with very narrow dorsal arms, until genitalia very curved downward in lateral view. That is completely different from other species of the tribe, when the genitalia shows basically the same pattern of variation. Yeah. So as I said, in this subgenus, the male genitalia have basically the same pattern, but the external morphology is quite variable. For example, eyes, pronotum, labium, abdominal pubescence in ventral view is quite variable, and each species group have different combination of these characters, making it very easy to identify the species. while in other subgenus, it is a more challenging. In terms of biogeography, we first proposed six biogeographic areas in the world based on the current distribution of the fish of the Bolshakova, and our best estimated model by Ribeiro, the DEX stratified model, indicates that the diversification of the Bolshakova occurred in the south portion of the New World after separation of Africa, in the areas C, D, and E, especially. The Mesoamerica, indicating the area F, is estimated as the most important area for the diversification of abilities and the Belostoma subgenus Tertostoma from phylogenetic events during the Paleocene and Eocene. The north of South America, indicated by letter E, is estimated as the most important area for diversification of lineage of Belostoma, especially those of subgenus Belostoma sensitive and Belostoma microbelostoma. As you can see here, in this clade, Belostoma plebeian group, Amazon seems to have played an important role in the initial diversification of this clade, and after some species expand their range of distribution to the northern areas or southern areas. Also, there is a tendency of species close related to share the same geographical area. As indication in this graph, species which diverged about 60 million years ago tend to show very, very high geographic autocorrelation index, indicating that the sister species or sister clades share the same geographical area. Even the specimens of those machines are good flyers, and maybe the dispersion is more restricted when we consider New World as a whole. In summary, we proposed for the first time a comprehensive dated phylogenetic hypothesis for those machines based on a combined data set. The subgeneric classification now is based on a phylogenetic hypothesis and supported by diagnostic characters, and divergence time and biogeographic history were estimated and discussed for the review. I'm grateful to many, especially my advisor during my PhD at the University of São Paulo, Eduardo Almeida, Andrew Short, who received me in Peru and supervised me during my internship at the University of Kansas, and Bob Tyts, who received me gently in his lab in Missouri, and loaned me very special material from Central America, who greatly improved my sampling. I am thankful to the other creators of visiting institutions and my funding actions. Now I'm here in Maranhão, and if you want to visit me and collect some tropical eruptions, you are always welcome. Thank you so much for your attention.
Right, thank you, Fabiano. We're kind of running out of time, so we actually don't really have time for questions. If you have any questions, perhaps you could email Fabiano directly. Thank you. Thank you, Fabiano.
Thank you. Thank you very much.
Hi again. A couple of things. Those who previously booked for the field trip but changed their minds, please let us know if they cancel the field trip. That would be nice to know if there is some people that for any reason finally does not come with us just to adjust in the restaurant and so on. And second, those who have colleagues who unfortunately haven't been able to visit us in Barcelona and can pick up the documentation for them and bring it home, that would be also very nice, because we have several bags with all the things you have. So maybe those who are at home would like to have them also, okay? Thank you, that's all. Let's go straight forward to lunch.